All right, let's dive right in. Today we've got a fascinating genomic detective story for you. It's about a critical gene that's been hiding in plain sight for over 500 million years, tucked away in, well, the last place you'd ever think to look. So let's crack this thing open.
So, what if I told you that one of your most important genes, a key piece of your biological instruction manual, wasn't filed away neatly on its own? What if it was actually written inside the pages of a totally different, unrelated instruction manual? That is the exact puzzle scientists found themselves staring at, and it's the mystery we're going to unravel today.
Okay, so this whole story revolves around serotonin. And yeah, we all know it as the feel-good chemical, the thing that helps regulate our mood. But here's the twist. Serotonin is ancient. Its story is way, way older than brains or nervous systems. It was playing fundamental roles in biology from the very beginning. And that ancient job, that's the key to everything.
To get to the bottom of this, we need to introduce our two main characters. And, on a genetic level, they are the absolute definition of an odd couple. They have seemingly nothing in common.
So on one hand, you've got the HTR2B gene. Its whole purpose is to build a little antenna for serotonin on the outside of our cells. It's all about communication. And then you have the PSMD1 gene, totally different gig. This guy helps build a cell's garbage disposal, its recycling and quality control center, so its job is all about maintenance. Two completely different worlds.
So, what's the connection? Well, it's not what they do, it's where they are. The HTR2B gene, the communicator, is located, get this, entirely inside a non-coding part of the PSMD1 gene, the maintenance guy. It's a gene within a gene. I mean, that's just bizarre. It's like finding the blueprints for a communications tower tucked inside a single brick of a recycling plant. Why would you do that?
The first question any detective would ask is, when did this happen? Is this just some random, recent evolutionary quirk? Or is there something much, much deeper going on here? We have to look at the timeline to figure out just how far back this strange relationship goes. And the answer is just mind-blowing. This setup isn't just in us, it's in all mammals. You find it in zebrafish, you find it in ancient lamprey, which is a jawless fish, you even find it in creatures that came before vertebrates, like sea squirts. We're talking about a genetic arrangement that has been perfectly preserved for over 500 million years. That is not an accident.
Now, to really appreciate how special this is, we need to talk about a huge force in evolution called whole genome duplication. Basically, think of it as a massive copy-paste error. Every once in a while, an organism's entire genetic library gets duplicated by accident. This gives evolution a spare set of every gene to play with, to create new tools without breaking the original ones.
Now check this out. Our evolutionary history has seen at least 3 of these giant duplication events. And other related serotonin genes? Oh yeah, they got copied all over the place, creating new family members. But this one specific nested pair? Nope. Every single time one of these events happened, the extra copy was lost. Evolution made sure to always go back to the original. It fiercely defended this one-of-a-kind structure.
So after all that evolutionary chaos, all those massive duplications, what's the magic number? 1. Nature went to absolutely extraordinary lengths to make sure there was only ever one copy of this nested gene. And when evolution protects something that hard, it means that thing isn't just a quirk, it is absolutely, fundamentally essential for survival.
Okay, so we've got our pattern, a 500-million-year-old structure that's been obsessively protected. Now we need the motive. What is this gene doing that makes it so indispensable? What makes it one of the body's master blueprints?
The answer takes us all the way back to the very first stages of life. See, in an embryo, the HTR2B gene is a master architect. It's helping build the stuff that forms your face and jaw. It is absolutely critical for building a healthy, working heart. It even helps get the blood system started. I mean, these are the most basic, non-negotiable steps to building a vertebrate body.
And you can really see just how vital it is when you look at what happens when it's gone. In studies of mouse embryos where this gene was turned off, the results were catastrophic. The heart ventricles were thin and weak. The liver, which should be busy making blood, was pale, indicating anemia. You take this one gene away, and the whole construction project just falls apart.
And here it is. This is the smoking gun. This quote from the research points out that if you knock out HTR2B's closest relatives, the HTR2A and HTR2C genes, these terrible developmental problems just don't happen. That proves it. The job HTR2B does is totally unique, it's ancient, and its more modern cousins just can't step in to do it. Okay, so we have all the pieces of the puzzle now. We've got the strange structure, the ancient pattern of it being protected, and a life-or-death motive. So let's put it all together and finally reveal this family secret.
Let's just walk through the logic one more time. The clue, a bizarre gene within a gene. The pattern, it's been protected for 500 million years. The motive, it plays an irreplaceable role in building an embryo. You put all that together and you can't help but land on one powerful hypothesis.
The big conclusion is that HTR2B isn't just another member of the serotonin receptor family. It's the ancestor, it's the patriarch, the original blueprint. The other receptors, like 2A and 2C, they probably came from those gene duplication events we talked about, and they evolved to take on newer, more specialized roles in the brain. But HTR2B kept its ancient, fundamental job as the master builder.
But you know how it is with all great scientific investigations. Solving one mystery just opens the door to another one, and this one might be even more interesting. The case is definitely not closed.
So we know why this structure was preserved. It's because HTR2B is just too vital to mess with. But we still have no idea if the structure itself actually does something. Why is this essential gene living inside a gene for the cell's recycling center? How do these two partners actually interact?
And this is really the next frontier for this research. Does being so close let them control each other? Maybe when one is turned on, the other is forced off? Do they compete for the same resources inside the cell? Or, and this is the really wild thought, is there some deep, undiscovered functional link between serotonin signaling and protein recycling? We know they're ancient partners, but the rules of their partnership, still a complete mystery.
And that leaves us with one last, kind of humbling thought. This whole incredible story, a 500-million-year-old secret about one of our most fundamental genes, was found hiding in a stretch of DNA that most people used to just write off as junk. It just makes you wonder. What other profound stories about our own evolution are just sitting there in our genome, waiting for the right detective to come along and finally crack the code?